second. I need to go back a couple, though. Okay, so I'm going to talk about the genetics of substance dependence and start with this slide. I think you'll probably see some variant of this slide in almost every talk for this meeting. And basically, it's to say drug and alcohol dependence are really important. They cause a lot of morbidity and mortality, and they're right up there ahead of a lot of medical disorders that receive a lot more productive attention in our society in terms of research and just attention from the public. Substance use is familial. So let's see, do I have a pointer? Not critical. Substance use is familial. This is, oh, brilliant, thank you. So this is a family of 3 generations of Chinese people enjoying a healthy, refreshing cigarette together. This is a poster that I bought in Hangzhou, China. And it's not just marketing. Use of various substances does run in families, leading to the most naive of questions: is it genes or is it environment? And no, it's not genes or environment, it's both. It's an interaction of genetic and environmental effects. Twin studies have shown that the heritability of most forms of substance dependence, all the ones that have been studied, is moderate, so 40% to 65%. So roughly speaking, that is the amount of the variance accounted for by genetic factors. So some are at the low end, like opioids, some are at the high end, like alcohol. But in any of these cases, genes are very, very important in determining who is going to be drug dependent and who isn't. So how do we know this? A key way is through twin studies. Identical twins share all of their DNA. Fraternal or dizygotic twins share half their DNA. So if a trait is influenced by DNA, identical twins will be more likely to share the trait than fraternal twins. So going back to the 70s and 80s, people did genetic studies, twin studies of many forms of substance dependence and demonstrated genetic influence. And in fact, you can go beyond showing that the disorders are genetically influenced to show in different parts of the lifespan, the influence of genes in red tends to go up, and the influence of family environment factors, which is a main kind of genetic influence, tends to go down. So early in life, when you're picking your peer group, the people you hang around with are very important. But as you age out of adolescence and into early adulthood, genes tend to take over and account for more of the variance. So what's the current status of our knowledge of genetics of substance use disorders? Well, substance dependence disorders have long been known to be genetically influenced for decades. There has been a lot of progress in identifying specific genes or risk alleles that influence risk just in the last decade. So why is that? It's been largely driven by newly available methodology like Genomewide association study, and by next generation sequencing and the associated statistical methodology, which has also grown up very, very fast with these new laboratory methodologies. So then I'm going to ask, what do you get from these studies? How are these studies confirmed? How can they be actionable? And then where do they go next? So just to get an idea of the dimension of the advance in the methodology, we can look at the progress in whole genome sequencing. So the first human genome sequence was completed only about 15 years ago, in February 2001. And that one genome cost $3 billion to complete. And there were 2 papers that came out more or less at the same time. They used slightly different methodologies. One came out in Science, one came out in Nature. But the total investment in research was about $3 billion in that one-person sequence. Turned out to be mostly Craig Venter's sequence, but that's another story. And that first $3 billion sequence was not even, by modern standards, a very high quality sequence. It was what was considered a draft sequence. It was more or less 90% accurate. So where are we now? This is a graph that shows the decrement in cost of sequencing technology. So down in this part of the slope, we have improvements in what are called Sanger sequencing methodologies. And here's the introduction of what's called next-gen sequencing, which is generally massively parallel optical sequencing. And when this was introduced, there was just an enormous decrease in cost, as there were a bunch of competing technologies and companies. In this sequence of 3 years, the increases in cost slowed down a lot because there was mostly one company. There wasn't a lot of competition. But now there's more competition again, and we have for 2015, we're approaching the $1,000 genome. So from $3 billion a genome to $1,000 a genome. So, you know, most people of moderate means, if you want your own genome and you want to know everything that's there, you can probably get it. So this is just an enormous change. The other parallel change is in genotyping technology, which led in part from the developments in sequencing technology because they allowed us to identify many points of inter-individual variation in the genome, which we call genetic polymorphism. So the basic plan of a genome-wide association study is you have a large group of patients or people who are ill or people with a trait that is not necessarily an illness, and a large group of people without that trait or illness. You genotype them simultaneously at, well, at the time of this slide in 2009, it was about 500,000 markers. Now it's more typically 1 million markers that go across the genome. And you look for statistical association of specific alleles that can be mapped to specific locations on the chromosome with the trait. This is a way to portray single nucleotide polymorphisms or SNPs across the entire genome. The chromosomes are portrayed linearly from chromosome 1, which is the largest, down to chromosome 22, which is the smallest autosome. There is a line that shows statistical significance in most of these plots. It's generally placed at the 5 × 10⁻⁸ significance value. That may seem rather stringent, but it's equivalent to about p.05 if you only do one comparison. The reason you have to choose a 5 × 10⁻⁸ cutoff is that you do so many comparisons and you have to do a correction for multiple statistical tests, which is sometimes called a Bonferroni correction. And then the things that float above the line are things that you want to investigate. And then generally there are follow-up studies that are necessary to prove that you landed in the right place. Now, this kind of portrayal of statistical results from a GWAS study is usually called a Manhattan plot, and this is why. It's because it looks a little bit like Manhattan, depending on the study. So a huge amount of progress in mapping genes for complex traits has been accrued by means of genome-wide association methodology. And this is a graph that shows each chromosome. So this, for example, again, is chromosome 1. And you can't read the chromosome labels, but this, for example, is 7, 6. And each of these colorful circles is a mapped complex trait that was mapped by a GWAS study. Almost none of these were known before the GWAS or genome-wide association study era. NHGRI, which is the US NIH Human Genome Research Institute, used to update this graph a couple times a year. They gave up in 2013 because it got to be too crowded and you couldn't really easily show it on one graph anymore. So we went from almost no circles on this graph in the mid-2000s to all of these thousands of GWAS findings. For example, if we look in the realm of psychiatric genetics and psychiatric traits, we can take the example of schizophrenia. For a really long time, and into the first GWAS that were done in the modern era, up to the late 2000s, with sample sizes in the thousands of subjects, there was nothing. And then there were a couple of risk loci. But now, with the advent of large meta-analysis studies, principally led by the Psychiatric Genomics Consortium, we know 108 statistically significant risk loci for schizophrenia. So this is the 5 times 10 to the minus 8th line. This is the Manhattan plot. This big peak here is in the HLA, also known as the MHC, major histocompatibility complex region. But there are all of these genes that with good statistical significance are now known to be associated genetically to schizophrenia, which has opened an enormously valuable window onto the biology and then the systems biology of the disorder. So a decade ago, this map of known risk loci for schizophrenia would have been nearly empty. And now, with the current knowledge, you can go practically anywhere in the genome and find a gene that influences risk for schizophrenia. So now I'll move on to some of the work that's been done in finding genes that influence risk for alcohol and drug dependence. And I want to make the point first that drug dependence genetics is by and large the same as what people refer to as pharmacogenetics. Pharmacogenetics is looking at the genetics of response to an extrinsic substance. So outside of our field of drug and alcohol dependence research, pharmacogenetics usually means response to an extrinsic drug that someone is using to treat a condition of some sort for the patient's benefit. But the ideas are similar for drug and alcohol dependence, and the principles are similar. So I'm going to go on to some findings for alcohol dependence. And by the way, Franz Schubert's glasses happen to be found in this city, but I do always use this slide for alcohol dependence GWAS. So let's first do the thought experiment. If we have to imagine the kind of gene variations that might lead to inter-individual differences in risk for alcohol dependence, what might they look like? Many people find genetic influence on drug dependence and alcohol dependence to be an intellectually difficult idea at first. And I want to show why that's not necessarily the case. So let's look at the main pathway of alcohol metabolism in humans. Alcohol is metabolized to acetaldehyde by means of the ADHs, the alcohol dehydrogenase enzymes. And this is actually a set of proteins encoded by a set of gene loci. Acetaldehyde is then metabolized by ALDHs, and the most important one in human is ALDH2, to acetate. Now, an interesting thing about this pathway is that we know about the pharmacologic effects of alcohol or ethanol, and by the way, ETOH, chemical symbol for ethanol or beverage alcohol. Many people in this room have probably taken a drink of alcohol on one occasion and know about that. And acetate really doesn't have much in the way of direct physiologic effects at all. There's often a fair amount of it circulating. But let's focus for a minute on acetaldehyde, the intermediary product. And that actually has some very interesting effects. High circulating levels of acetaldehyde are poisonous, and they lead to what's known as the flushing reaction, characterized by facial flushing, lightheadedness, palpitations, and nausea. They are physically unpleasant to the person who experiences them. So besides being physically unpleasant, high levels of acetaldehyde can increase risk for certain GI tract cancers. So it has a lot of importance in various kinds of pathophysiology. Acetaldehyde is a metabolic byproduct that sometimes hangs around in the body for a pretty long time. There are different ADH enzymes that vary in the speed and efficiency with which they metabolize ethanol. And there are also variants of ALDH2 that interfere with the clearance of acetaldehyde that result in changes in the amount of acetaldehyde that's circulating in different individuals. So the flushing reaction, some people, particularly people of Asian ancestry, tend to flush when they drink alcohol. And this is associated with rapid heart rate, sometimes also with nausea and headache. This is mostly genetically determined, and it's perceived as aversive. So this is just a picture of a guy, an Asian guy who took a drink of alcohol and posted a picture of himself on the web having a flushing reaction. So going back to the metabolism of alcohol, if we continue our thought experiment, if we knock out the function of this protein by knocking out the function of the gene, then ethanol will be metabolized to acetaldehyde, and then the acetaldehyde will just hang out there. And that can cause this flushing reaction. So now I'm going to describe some results from a study that we did in a Chinese population. We recruited a sample of 600 subjects in northern Hunan province. We interviewed them with the Diagnostic Interview of Genetic Studies, known as the DIGS, and we genotyped them with a sparse GWAS array. And this is the Manhattan plot showing the results from that study. And you can see that there are 3 traits portrayed in this slide: alcohol dependence on top, maximum number of drinks, and this is the flushing response itself. And in all cases, there is a significant signal mapping to chromosome 12 at ALDH2, the alcohol metabolizing enzyme. The greatest statistical significance is with flushing response, and that's at about 10 to the minus 26th. So that's a very, very significant finding. The next highest significance is with maximum drinks. So maximum drinks is a trait defined as the answer to the question, what's the largest number of drinks you've ever consumed in any 24-hour period in your life? It turns out response to this one question is just about as heritable as alcohol dependence itself. So we have here genetic variation at this alcohol metabolizing enzyme, ALDH2, greatly influencing the maximum number of drinks that people can take in their lifespans. And finally, we have a significant association with alcohol dependence itself, although less statistically significant. Now, we did a plot of the subjects in this study by ALDH genotype, and we talk about different alleles at a polymorphic locus. So these are just the genetic bases that the people have on their 2 DNA strands. So for this variant, RS671, A is a protective allele and G is a risk allele. So if you do have 2 copies of this protective allele, you flush, but also your most likely bin for maximum number of alcoholic beverages consumed in any 24-hour period is 0 or 1 drink. That's your most likely circumstance. Whereas if you have no copies of the protective allele, the most likely bin you fall into is more than 25 drinks in a 24-hour period. So this is a rather large genetic effect caused by a single base change out of the 3 billion in the genome. And it's really pretty easy to understand how this base change could affect propensity to become alcohol dependent. So why is it? If you have the protective allele, then if you take a drink of alcohol, even a single drink, you flush and you feel kind of sick. So people having some rational aspects to their behavior, if they become sick when they drink alcohol, are less likely to drink a lot and are less likely to become alcohol dependent. And I really like this as an illustration of genetic influence on substance dependence traits, because you don't even have to get into reward systems and psychological mechanisms. It's very simple single base change. If you have the protective allele, then if you drink alcohol, you get sick, therefore you're less likely to get alcohol dependent. So now I'm going to discuss a bit of a study that we did in the United States population of alcohol dependence. And this study is one of many results that we got from a series of studies funded by the National Institute on Drug Abuse and the National Institute on Alcohol Abuse and Alcoholism. We started with affected sibling pair linkage. We continued with case control association and GWAS. The leaders of the study were myself at Yale and Henry Kranzler, my co-PI, who's now at University of Pennsylvania. It was a large collaborative study that spanned 7 R01 grants and an RC2 grant, resulted in collection of a lot of data, and I'm going to present results from a couple of studies that derive from this sample collection. So first I'm going to go straight to the Manhattan plot for alcohol dependence in African American subjects in our sample. And you can see here's that 5 times 10 to the minus 8th significance line. And there is one finding that's very highly significant here that gets to better than 10 to the minus 12. I'm not going to show the slide for European ancestry subjects, but it actually looks very similar to this one. So I said previously ALDH2, aldehyde dehydrogenase, is on chromosome 12. What's on chromosome 4 that leads to these signals in European and African ancestry subjects? Well, it turns out that it is a gene that encodes another protein involved in alcohol metabolism. In this case, it's ADH1B. And in European American ancestries subjects, it is a functional variant that gives rise to an ARG48 his protein change. In African Americans, it's a completely different polymorphism with a completely different protein change, an ARG to cis change. In both cases, the minor alleles for the genome, or the less common alleles for the genome-wide significant hits, are protective against alcohol dependence. So where does this sit with alcohol dependence? Well, in Asian populations, it's a variant in ALDH2 that interrupts the metabolism of acetaldehyde that's protective. In European and African ancestry subjects, it's different variants of ADH1B that, as it turns out, the functional biology has been done, speed up the metabolism of ethanol to acetaldehyde. In both cases, it leads to increased amounts of acetaldehyde circulating in the bloodstream and an unpleasant physiological reaction that's aversive to the person who drinks, and therefore results in less alcohol dependence. So again, if we look at the top GWAS, Genome-Wide Association Study, alcohol dependence risk SNPs, in European and African ancestry, it's completely different variants, but in the ADH1B gene. In Asian ancestry, it's a functional variant in ALDH2. You might ask, why are they different variants in European and African ancestry samples? Well, it seems to be just an artifact of population history without any particular meaning so far as we know, other than that the protective variant in European Americans is absent in African subjects, and the protective variant in African ancestry subjects is absent, does not even occur in European ancestry subjects. So we have the development of convergence in terms of pathway, sometimes even for the same gene, but not the same variants in 3 different populations. So now I'm going to talk about some genetic findings in other substances of abuse. There are extant findings for nicotine, opioids, cocaine, and cannabis. I'm going to talk briefly about nicotine and cocaine. Nicotine dependence, legal almost everywhere, is really the best studied of the drug dependence traits, because biologically nicotine is a drug. And why is that? It's because many studies of medical traits like cardiac function, diabetes, pulmonary function include at least a rudimentary assessment of cigarette smoking. because it's a very widespread and well-recognized health hazard that is known to impinge on a lot of other medical traits. So if someone is collecting 20,000 subjects for a genetic study of cardiac function, then chances are there will be at least some smoking data collected too. And this has resulted in a wealth of data for association studies of nicotine dependence. The landscape for nicotine dependence is interesting because there have been generally consistent findings in many, many studies showing associations with variants in one particular region of chromosome 15 that harbors 3 nicotinic receptor genes, CHRNA5, CHRNA3, and CHRNB4. These encode the nicotinic alpha-5, alpha-3, and beta-4 receptors. So this goes back to 2010, when in a single month there was a Nature paper and a couple of Nature genetic papers. And this is another Manhattan plot, 5 times 10 to the minus 8th line way down here. And this is a log scale, so it's 27 orders of magnitude better than that. Association with CPD, that's cigarettes per day. It's somewhat analogous to the max strengths alcohol dependence measure. It's the number of cigarettes a subject habitually smokes per day. And you have this huge signal in this chromosome 15 region that harbors 3 nicotinic receptor genes and very little else in the whole rest of the genome. And similar finding in one of the other studies that came out in the same issue of Nature Genetics. So why does this make sense? Well, these are 3 nicotinic receptor genes. So it's a good illustration of pharmacogenetics. Nicotine is the drug, and one of their main points of interaction with behavior and brain function is with the nicotinic receptors, the receptors that can respond to nicotine. So it really makes sense for genetic variation that affects the function of some nicotinic receptors also affects the perception of nicotine by the individual. And there's actually enough data for the effect of each of these risk alleles to be quantified. So in a subject who smokes, who is nicotine dependent, each one of these risk alleles that you get accounts for an additional roughly 0.85 cigarettes per day. So if you have 2 risk alleles and you're nicotine dependent, you smoke on average about 1.7 more cigarettes per day than someone who's nicotine dependent and does not have these risk alleles. So it makes sense, it's easy to appreciate, but these particular nicotinic receptors were not considered key candidates prior to these genetic studies. They are not the most abundant nicotinic receptors in brain, and prior to the GWAS or Genome-Wide Association studies, more bets would have been on alpha-4 and beta-2 nicotinic receptors. So the genetic studies led to a lot of insight into the pathophysiology of inter-individual variation in nicotine dependence risk. We did a study on these alpha-5 variants too, and we took it in a slightly different direction. We looked at interaction of childhood adversity with these now known risk variants and nicotine dependence risk. And we found a difference by sex, and we found that the effects of the risk variants were much greater in male subjects in the context of childhood adversity than without childhood adversity. And we're going to hear more about this kind of thing later in the meeting. This is one example among many where early life experiences play a big role in later life risk for substance dependence traits, in this case, interacting with a genetic risk factor. I'm going to talk also briefly about cocaine dependence. There's less to say about it. So far, there's only one GWAS, which came from my group. There are no replication samples because no one has funded collection of the samples that are needed to do the study beyond our initial sample, which is a good illustration of the lack of appreciation among funding agencies worldwide for the importance of getting to understand the genetics of these traits. So our study did have genome-wide significant findings. Are there other ways to confirm these findings without independent replication samples? So first of all, this is a regional Manhattan plot from our study. This is focused in, it doesn't show all the chromosomes, it just shows the chromosomal region with the genome-wide significant finding. In this case, the significant finding maps to a gene called FAM53B. This is not a gene that had been studied much before, and it's certainly not a gene that had been implicated in cocaine dependence. In the absence of a replication finding, how do we know if this is a new window into the biology of cocaine dependence or something else? It could be a false positive finding. Well, there are some new results that can help us understand this a little bit better. This goes back to a 2005 linkage study, which is another kind of genetic study that we did that was just giving chromosomal locations for risk genes, also landed in the FAM53 region. But a mouse paper just was published on November 19th, and this was a genome-wide association study in a mouse model of cocaine self-administration. And that study had 2 genome-wide significant findings, and one of them mapped to exactly the same gene of little-known function that our human study landed on. So signals now in the 2 different species, mouse and human, give pretty good support to FAM53 being a gene that's worthy of a lot more study as we go to understand the biology of cocaine dependence. Before our GWAS study, the best candidate genes or places that people would have guessed might be involved in risk for cocaine dependence would probably have involved dopamine metabolism or the dopamine reuptake site, the dopamine transporter, and certainly not FAM53B. Genetic studies were needed for this insight. The last thing I'm going to talk about is a little bit about clinical translation and how some of these findings may get to be of use in a clinical context. Back in 2009, my colleague Henry Kranzler published this genetic association of alcohol dependence where he studied the gene for GluR5. The GluR5 is the name of the protein, GRIC1 is the name of the gene, and demonstrated genetic association of variants at GRIC1 with alcohol dependence. So interesting finding, not highly significant, might have been filed away. Then Kranzler went on to study topiramate in a treatment trial, and topiramate is an emerging treatment for alcohol dependence. Topiramate, it turns out, has major action on the protein product of the GRIC1 gene. So Kranzler and colleagues decided to examine whether genetic variation at the GRIC1 gene, a major site of action of topiramate, could influence clinical response to the treatment. So first he did the treatment study itself, where he compared placebo and topiramate and showed that topiramate has a robust effect on reducing heavy drinking days per week, and also other measures of response to treatment. For example, it also increased the number of abstinent days per week. So this is very good, but there had been a couple studies before that had shown that topiramate is a good treatment for alcohol dependence. But remember, topiramate acts on the receptor encoded by GRIC1. So Kranzler then asked the question, does this GRIC1 variation have an effect on response to topiramate? Because if it did, then you could do a genotype test and you could know something before starting treatment about if a subject would respond or not. And what he found was that for some subjects, GRIC1 genotype is indeed highly predictive. So if you take a look at subjects with 2 copies of the A allele, or people with 1 copy of the A allele and 1 copy of the C allele, Topiramate and placebo were statistically not distinguishable in terms of their effect. It's only for subjects who had 2 copies of the C allele at this GRIC1 locus that there was a very robust treatment effect of Topiramate. So this is an example of a genetic finding being translated into clinical utility and creating a situation where it appears that by doing a genotype test, we may be able to identify those subjects who are likely to respond to this medication. And then this could help us allocate subjects into a good personalized treatment, what some people call personalized medicine based on genotype. This needs replication. It should not be regarded as established clinical science yet, but it's a good study. So implications and next step. You can actually learn a lot about GWAS and sequencing studies beyond the risk alleles that hover above that 5 × 10⁻⁸ line. You can consider a lot of the rest of the output from GWAS studies. You can use it for pathway analysis. So for example, our pathway analysis from our opioid dependence genome-wide association study identified as the most important pathways for genetics of the trait to be not opioidergic or dopaminergic transmission as we might have predicted, but rather calcium systems and potassium signaling. Somewhat counter to expectations, and also places opioid dependence in the realm of a host of other psychiatric traits like bipolar disorder and schizophrenia, where calcium signaling has been shown to be important. These findings could be important for drug discovery, prediction of drug response, and identification of what might be called druggable protein targets. We can compile the results of GWAS into what are called polygenic risk scores, which are useful for risk prediction. Not yet in the clinical arena with current state of knowledge, but very interesting in the research arena at this point. And we can also use GWAS data to investigate overlap with other disorders. We have started to do this with substance dependence traits. We have some papers submitted and under review, but nothing published yet. So finally, I would like to acknowledge support mostly from NIDA and NIAAA, and acknowledge the contribution of many collaborators, most importantly, Henry Kranzler, my collaborator for over 2 decades now. I will also point out that due to NIH sharing policies, GWAS data must be shared and made available to the public. If anyone wants to do additional analyses or just look at my GWAS data that I described, it can be downloaded from dbGaP, which is the NIH data sharing site, after completion of a short application. And that's my research group. And with that, I would like to thank you for your attention.